The cell cycle is a series of events that controls the growth and division of a cell. The part of the cycle when the cell divides is called M phase, and the rest, when the cell is living, is called interphase. Mitosis is a process in which eukaryotic cells replicate into two identical daughter cells with identical genomes. This is an example of asexual reproduction, which means the daughter cells are identical to the parent cell. Mitosis helps a multicellular organism grow and repair itself, and it helps unicellular organisms replicate themselves. Mitosis is a multi-step process with 4 phases known as prophase, metaphase, anaphase, and telophase. There is also a correlated event called cytokinesis, which happens around the same time during M phase.
Interphase. There are 3 phases to interphase: gap phase 1, the synthesis phase, and gap phase 2. During gap phase 1, the cell grows and produces many proteins and organelles. The cell has just replicated, so it needs to produce organelles like the Golgi apparatus, the endoplasmic reticulum, and ribosomes. Different cells spend different amounts of time in gap phase 1. Some cells reproduce very slowly or not at all. This is called gap phase 0. This happens when a cell isn't signaled to pass the checkpoint by a cyclin-dependent kinase. The cell is performing its normal function but isn't getting ready to duplicate. A cell can be launched back into the cell cycle by some sort of stimulus. The synthesis phase. During the synthesis phase, a cell continues to grow, but it also duplicates its DNA. The chromatids are duplicated into sister chromatids, which are connected at a centromere to form chromosomes. Gap phase 2. During gap phase 2, a cell is growing and producing proteins just like in gap phase 1. The DNA in the nucleus has already been duplicated, and the cell is approaching mitosis. A lot of ATP is being produced during this phase.
Now, on to mitosis, starting with prophase. During prophase, the mitotic spindle fibers begin to form outside the nucleus. The centrosomes begin to move to opposite poles of the cell. The sister chromatids begin to condense into chromosomes, their chromatin becoming denser, and the nucleus begins to dissolve, preparing for metaphase. The nucleus is fully dissolved by metaphase. The mitotic spindle fibers begin to attach to the chromosomes. These chromosomes are then pulled back and forth before finally settling in the center of the cell, creating a planar region cutting the cell in two. This is the metaphase plate. Anaphase. Once all of the chromosomes have aligned, the centrosomes pull the chromosomes apart, one half of each chromosome ending up towards one of the centrosomes. This is how each daughter cell receives the same genome as the parent.
Telophase starts when 2 identical nuclei appear from the remnants of the first nuclei. The chromosomes become disordered when they are removed from the spindle fibers. Then the spindle fibers break down. Cytokinesis begins when the cleavage furrow forms. It forms by both membranes being pulled apart, which leads to the even splitting of the cytoplasm. In a plant cell, the cleavage furrow does not form, but a cell plate forms in the center.
Final reflection questions. Question 1. How could you modify your model to better illustrate each process if you were to perform this simulation again? To make this model more accurate of mitosis, we would show what interphase looks like in a cell. We covered interphase with the voiceover, but we could have done a better job with depicting interphase in our model. We could have also shown the cleavage furrow more accurately. The way we depicted it, it looked like cytokinesis was already almost complete during telophase, which is inaccurate. Question 2. What did you learn about cell division through this modeling activity? In this activity, we learned about centrosomes, which contain the centrioles, and how the spindle fibers form from the centrioles. We learned about how cells regulate the cell cycle automatically through checkpoints, which require a specific chemical signal to pass. We learned how the spindle fibers attach to the kinetochores of the chromosomes to pull on them. Lastly, we learned about some cells that are stuck in gap phase 0 and don't continue past the gap phase 1 checkpoint. Cells like this require an external chemical messenger to allow them to pass the gap phase 1 checkpoint. Without that chemical messenger, they will perform their job in the human body, but they will not divide.